We have learned the diversity in the configuration, biochemical processes, and microbial communities of the biological processes used for wastewater treatment and resource recovery. Now, we will start to unify these diversities with the fundamentals. In this class, we will establish a basic concept of energy flow in biological reactions. One of the commonality in biological processes is the flow of energy. The electron donors contains reducing power, which can be used to generate energy. When it reacts with the electron acceptor, energy is released via oxidation-reduction reaction. Such energy is used for cell growth and cell maintenance. In some cases, like during anaerobic digestion, the reactor end product, methane gas, can be collected for energy recovery. The release of energy during chemical redox reaction is vigorous and visible due to the instant drop of energy level. The electron donor, in this case the couch, reacts with the electron acceptor, oxygen, and releases energy in the form of heat. For biological reactions, this is too explosive. So instead, in biological reactions, energy is released in a more controlled manner. It is passed down stepwise to multiple intermediates by electron and energy carriers. By regulating the amount of these carriers, the flow of energy is controlled. A typical electron carrier is nicotinamide adenine dinucleotide, NAD+, and its reduced form NADH. NADH can be used to generate a common energy carrier, ATP. To generate ATP, NADH transports electron to form a proton gradient outside of a membrane, creating an osmotic pressure. Due to the osmotic pressure, the proton flows back in through the ATP synthase and drives the production of ATP from ADP. And ATP can be used as an energy currency to drive various biochemical reactions, such as cell synthesis and cell maintenance. In the last class, we mentioned that one of the major biochemical processes is the aerobic degradation of carbohydrates. This is a fairly detailed stepwise degradation of glucose. It is first degraded to acetyl-CoA, a coenzyme A complex of acetic acid, which is a common intermediate that participates in many biochemical reactions. It can also be produced during the beta oxidation of fatty acids. Acetyl-CoA then enters one of the most important and common metabolic pathways, the citric acid cycle, or Krebs cycle. As you can see, electron carriers like NADH and FADH2 and energy carriers like ATP is generated during this process, which provides energy for various cell activities. But where is oxygen in this aerobic process? Oxygen is used to regenerate NAD+ from NADH. We can write out the half reactions. NADH donates electron and forms NAD+, and oxygen accepts that electron to form water. We can see where each of the half reactions stands on the energy level chart. Because we have flipped the top reaction and write it as NADH donating electron, so the sign of the free energy change, delta G, becomes negative as electron is lost. Combining this half reaction, we can get the overall reaction and the overall free energy change. This chart shows the stepwise decrease of energy level of the intermediates involved in glucose degradation. Under anaerobic conditions, the degradation pathway changes. Instead of being converted to acetyl-CoA, it stops at pyruvic acid and can go through several different options afterwards. The pyruvate can be converted to lactate, acetate, butyrate, and butanol, all of which can participate in the following reactions such as cell synthesis and methanogenesis. Compared to the aerobic degradation, much less electron carrier and energy carrier are produced from 1 mole of glucose in anaerobic pathways because of the high energy state of the end products. So the anaerobic process is not as efficient in transferring energy to support cell growth and maintenance. If we look at the free energy production from the anaerobic regeneration of NAD+ quantitatively, pyruvate is the electron acceptor instead of oxygen. Much less free energy is released overall from the oxidation of NAD+ anaerobically. In summary, anaerobic process produce less NADH, and each NADH can produce less energy when oxidized anaerobically. We can expect a much lower growth rate and yield from anaerobic microorganisms compared to the aerobic ones. Energy generated from catabolism, the breakdown of electron donors, is used for anabolism, the biosynthesis for cell materials. Anabolism is essentially the reverse of catabolism. The NADH and ATP produced from catabolism are used for biosynthesis. Based on that, we can write down the energy needed to produce glucose from acetate and CO2, which represents the heterotrophic and autotrophic glucose synthesis. More free energy is needed in autotrophic process as carbon source is at a lower energy state. The reaction of synthesizing the empirical biogas from glucose is shown here. To summarize, in catabolism, the electron donor reacts with the electron acceptor to generate end products. Its reducing power reduces NAD+ to NADH, and its energy drives the conversion of ADP to ATP. NADH can also drive the ATP production. ATP then can provide energy to anabolic reactions that convert the carbon sources and nitrogen sources to cell biomass. ATP is also used for cell maintenance like motility, active transportation, antimicrobial resistance, etc. When there is abundant substrate and low biomass concentration, most of the energy from the electron donor is used for cell growth. When the substrate concentration is low and the biomass concentration is high, most of the energy is used for cell maintenance.